Well, thank you so much. It's a pleasure to be here. You know, I really enjoyed all the lectures. This morning, hearing people talk about their careers, I thought I'd start with mine, which was far less auspicious than theirs. But just to give you an idea of what I did. So I got my degree in chemical engineering from MIT in 1974. And that year, there was this gas shortage in the United States. And actually, if you lived in Boston, where I did, you actually had to wait in line for 2 hours to get your car tank filled up. But the consequence of that is that if you were a chemical engineer, you got a lot of job offers. And so all my friends that year, they went to work for oil companies. I thought I'd do that as well. So I applied to oil companies, and I actually got 20 job offers, 4 from Exxon alone. And I wasn't very good, but they just had so many openings. But one of them made quite an impression on me. When I went there, they said, gee, if you could just increase the yield of this one petrochemical by some tiny percentage, they said that would be great. It would be worth billions of dollars. And I remember flying back to Boston thinking to myself that I didn't want to spend my life doing that. Made me think what I did want to do. And when I was a graduate student, I spent a lot of time helping start a high school for disadvantaged high school kids in the Cambridge area. And one day, I saw an ad for being an assistant professor at City College of New York to develop a new chemistry curriculum, which is largely what I did at that high school. And so I wrote them a letter applying for a job, but they didn't write me back. But I liked that idea, so I found all the ads I could to do something like that. I found about 40 of them, and I wrote to each of them, and none of them wrote me back. So that wasn't going so well. So I started to think, well, what other way could I use my chemical engineering background to help people? So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But one day I remember walking into the lab, and one of the older scientists said to me, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. He thought very, very highly of Dr. Folkman. I won't say what he thought about me. At any rate, I wrote to Dr. Folkman, and he had this idea, and he was kind enough to actually offer me a job at Children's Hospital. Didn't pay like the oil companies, but I thought it was very important. I knew nothing about biology then, but basically his idea was shown here about how a tumor grows in relation to its vasculature. And he had this theory that tumors made a chemical signal called TAF, or tumor angiogenesis factor, that would diffuse out to the surrounding blood vessels, which normally were quiescent. But in the case of the tumor, the vessels started growing to the tumor and feeding it, and it would keep growing and ultimately metastasize through these vessels, as was discussed earlier this morning, and that's often what kills people. So his theory was that if you could stop the blood vessels from growing, that might be a whole new way to treat cancer. Now, he was widely criticized for this theory. At any rate, he hired me to prove he was right, and in so doing, to isolate the first angiogenesis inhibitory factors. Now, one of the biggest problems whenever you try to do something like this is you have to have what we call a bioassay to study the blood vessel growth. And there wasn't any at that time, and it's not so easy to do because there were always background blood vessels everywhere, except for a few places, but what's called the chick-chorial-ontoic membrane, where there are very few, and actually the cornea, where there are none. And what we talked about is maybe we could put certain tumors in the cornea, which would cause vessels to grow, but then we needed a tiny delivery system, like a microparticle or a nanoparticle, that could deliver the molecules and extracts that we were isolating, all of which were quite large molecules, like proteins and things like that, nucleic acids. And delivering them from a tiny particle, that had never been done before. In fact, in 1974, when we were doing this work, the idea of what I'll call an advanced drug delivery system, hardly anybody had worked on that. There was only one company actually in the world doing that, a company called Alza in Palo Alto, California. And Dr. Folkman was on their board, as was a number of Nobel Prize winners in chemistry and other areas. And he went out and he asked them, could they help us? But what they said was that you really can't do that. The molecules are too big to be delivered from these materials. It's kind of like maybe walking through a wall. At any rate, if we we're going to solve the blood vessel problem, we had to solve this problem. So I spent several years in the laboratory experimenting with different materials, different designs, different techniques. I actually found over 200 different ways to get this to not work. But eventually, we were able to make little microparticles or nanoparticles. And we see a picture of one and one cut in half. And in 1976, we published in Nature the fact that you could use this kind of approach to deliver all different kinds of proteins. And it was the first paper, to my knowledge, showing you could deliver nucleic acids, for reference, DNA or RNA or nucleic acids. So I want to go back to 3 months before this paper came out. I got asked for the first time in my life to give a talk at a major meeting. I was 27 years old, and I practiced this talk for weeks into what's called a tape recorder. And finally, the day came, and I got up and I gave that talk, and I thought at the end of it, I didn't do that badly. You know, I didn't forget too much what I was going to say, or I didn't stammer too much. So I thought when I was done with the talk that all these older scientists, being nice people, that they would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, well, we don't believe anything you just said. And they would repeat that the molecules were too big, and also the solvents like methylene chloride and ethanol would destroy whatever we put in. And things actually went downhill for me from there. I tried to get research grants. My first 9 were rejected. You know, people said that an engineer knows nothing about biology or oncology. So that wasn't going so well. Also, you know, I liked being a postdoc at Children's Hospital, but a lot of my friends said probably you should apply for faculty positions at some point rather than do this your whole life. So I did. I applied to faculty positions to chemical engineering departments, but no chemical engineering department in the world would hire me. They all basically said, well, I should, if you're trying to do bio stuff, that doesn't make any sense for a chemical engineer. You should do energy or oil or mathematical modeling. So I didn't get any jobs. Finally, I got a job in a nutrition department. The reason I think I got it was the man who was the head of that department, Nevin Scrimshaw. He was what I'll call a benevolent dictator kind of department head. Meaning he liked me, so he offered me a job, but he didn't ask anybody else in the department what they thought. Which would have been okay, except for one thing, which is the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. And I'm not just making it up. Actually, Mike Marletta, who's a very well-known chemist, was head of Berkeley's chemistry department, member of the National Academy of Science. He was actually on the MIT faculty in 1979, as well as myself. And I'll just, number of years ago, I received some award, and they asked him to say a few words about what it was like to be with me. I'll read you some of it. He said, one evening, I went to a faculty dinner at a Chinese restaurant with me and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So like I say, my first years at MIT were not that great, but I'm still there, as Laurie mentioned. But the good thing is that since we developed these delivery systems, we could now study this angiogenesis problem. So what I'm going to, again, people, if you don't like the sight of blood, you shouldn't look, but basically I'm going to show you rabbit eyes from a paper we wrote in the journal Science. And what you'll see is, this was a 1976 paper, is that if you and we probably did like 2,000 different eyes on the left, and you always get this pattern, and you can even measure the length of the longest blood vessel, and that's with a control. It's also with, we had actually lots of controls because almost everything we isolated didn't work. But then if you have one fraction, it wasn't purified. We repeated this one 21 times on the right, and you can see that the vessels are sparser, they're lower, they even avoid the tumor. So this showed, we like to think, for the first time that blood vessel inhibitors did exist, but it would also provide a pathway for all future inhibitors to be tested and found. Now, medicine, as you imagine might know, even from some of the talks today, it doesn't move that fast generally. And it actually took 28 years and literally great work from a company, Genentech and others, to isolate the first, purify and develop and get FDA approval for the first inhibitor. And this actually is Avastin. This may be hard to see in the back, I don't know. But Avastin is, if you look at it, it's one of the top best-selling drugs in history. And not only did that get used, and actually a number of angiogenesis inhibitors still get approved in 2024, and they're used for many kinds of cancer, but they're also actually pretty much the only drugs that can be used today for treating diseases of blindness in the back of the eye, diseases like macular degeneration or diabetic retinopathy. At any rate, I want to go back to the 70s again. So Children's Hospital is in Boston, it's a pretty good hospital, but in 1974, it had 0 patents, and Dr. Folkman and I talked about this, and we thought, well, let's try to file a patent. So we did, first one there, and actually it got rejected 5 years in a row. And I remember the head of the tech transfer program coming to see us one day, or see me, I guess, and said, Bob, you are wasting so much money. He said, you have to give up. It's never going to get allowed. But I don't like to give up. So I started to think, how could we convince the patent examiner to allow this patent, legally, of course, and science really wasn't working. So I thought, as I told you in the beginning, when we first started doing this, people said it was impossible, you couldn't do it. I wondered, maybe somebody wrote that down. So in 1982, I did a science citation search on our 1976 Nature paper to see what I might find. And I found some interesting quotes, but this one I liked. This was from 5 of the top material scientists in the world then, and they just wrote about the agent to be released as a relatively small molecule with a molecular weight no larger than a few hundred. One wouldn't expect that macromolecules could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So I showed this to the people at the hospital, and they flew down to Washington to show it to the patent examiner. And he said, well, he had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits, written statements from these 5 people that they really wrote that. So it's a true story. So I wrote them, and they were all nice enough to write me back. They really did write it. And we got this very broad patent allowed. And, I'd been working on this now for about 10 years, and I always had this dream that something that I do, might make its way to help people someday. But, people cited our work, as Laurie mentioned, but I really want to see it get used. And of course, you can't, you probably need a company to do that. So finally, after 10 years, I got a call from one of the leading animal health companies who are interested in using this for animal health molecules, and then the next year from a leading human pharmaceutical company that wanted to use it for humans. And they said, well, gee, we're going to give you a consulting fee, we're going to give you a big grant, and most important to me, they were going to work on it. And I thought this was great, but they each worked on it for about a year, and then they gave up. And so that wasn't so good, even though I did get a better car. But at any rate, so then Alex Klebanoff, one of my friends, said, Bob, we should start our own company. So we started a little company. Actually, Alan first was kind enough to join the advisory board. And through that company and other companies we started, actually many products would get developed, and our students would work at these companies. I'm just going to show you some pictures, but basically we don't have to go into detail, but they're used for all kinds of products, probably by hundreds of millions of people every year. And not only are they used in human medicine, but even they're widely used in the area of aquaculture because of work that Yanni Zohar started doing when he came in our lab, which would be little implants to control fish spawning and actually even preserve rare fish. So that's kind of how I got started. It took a while, but at least eventually something happened. But I did notice when I was working at the Children's Hospital, I started working on materials. In fact, that's the title that Shu Gang gave me today. And when I was working there, I was curious, how did materials find their way into medicine? Was it by chemists or chemical engineers? And I thought, that must be the case. But almost always what I found, that the driving force for getting materials into medicine was medical doctors. And what they'd almost always do in the 20th century is they'd go to their house and find some object that kind of resembled the organ or tissue they wanted to fix, and then they'd use it in a person. And specifically, like artificial heart, that was some surgeons, heart surgeons, wanting to make an artificial heart. And they said, well, what object has a good flex life? And they thought about a lady's girdle. And that's actually what they made the artificial heart out of. But if you follow this, the artificial heart hasn't worked real well. One of the reasons is that the blood hits the surface of the artificial heart, the lady's girdle material, forms a clot and goes to the patient's brain. They get a stroke and they may die. But if you think about it, something that was designed to be a lady's girdle, it may not be the optimal blood contacting material. So at any rate, see, the issue is, like this was started in 1967, but once you start down that path, it's not so easy to change materials from an FDA standpoint. So that's actually still what the artificial heart is made out of today. And this problem pervades all of medicine. I mean, breast implants, for example, one was a mattress stuffing. So I started thinking, I'd learned chemical engineering, and I thought, well, maybe rather than go to your house, what if you ask the question, what do you really want in a biomaterial from a chemistry, engineering, and biology standpoint? And then could you synthesize it from first principles? So we picked an example, and I'll just show you one. We do a number of these. But when we started in 1979 on some of these things, the only material that was approved by FDA that was synthetic and degradable were the polyester sutures. They would dissolve, as you see here. It's what we call bulk erosion. Over time, water creeps in and you get holes, and ultimately it breaks apart, which is fine for a lot of things. But for others, you could imagine if you had a drug like insulin or an anti-cancer drug, it might dump out, and that could be toxic. So we said, from an engineering standpoint, what we'd really like is this, surface erosion, kind of like the way a bar of soap dissolves. And so we tried to ask, well, how could you do that? So from a molecular standpoint, if you were using a polymer, which is what we were thinking about, polymer is monomers, like polyethylene. The monomers are all ethylenes. And polyesters, which a lot of these were before, the bonds are ester bonds. So we started asking questions, but design questions. That's what chemical engineers do. First question is, well, what should cause the degradation? Should it be water? Should it be enzymes or something else? Well, everybody in this room probably has different enzyme levels, and the tissue response around a material will change over time. So we thought that's not going to be so reproducible. But all of you have excess water. So we said, let's make this so that water degrades it. But then you go to the next question. If you do that, how do we make sure the water doesn't creep in and cause that bulk erosion? So we wanted to make these monomers what we call very hydrophobic or water repellent. And then we said, well, let's go to the next question. If we do that, how are we going to make it degrade fast enough? So now you could go to organic chemistry and say, well, what bonds might make it dissolve fast enough? And we estimated going up to the top of that, which is the anhydride bond, and therefore proposed the idea of synthetic polyanhydrides. And of course, and then I got people like Mike Marletto, who is a chemist and also toxicologist, and others to help us and say, well, what of these building blocks might be safe, and yet we could still make good polymers out of? And we came up with this structure, which we put on the blackboard. But the interesting thing about this is what we call a copolymer. And a copolymer, by adjusting the ratio of, say, the CPP to the safe basic acid, we thought, well, you could adjust how fast this will degrade. But still, we thought it might show close to surface erosion. And it actually did. If you just change the ratios of those, 79% of the one, it's all gone in 2 weeks, but 0%, it's like 8%'s gone over 14 weeks. It'll actually take 3 or 4 years for a millimeter thick slab to dissolve. So again, one of our goals, again, maybe it's because I'm a chemical engineer, I wanted to do the see that we could prove this principle, but I thought I'd love to see it be used for something good. So Henry Brehm in 1984, he was a neurosurgeon. Today he's a chief of neurosurgery at Johns Hopkins, but he came to see me and he was interested in coming up with a new way of treating brain cancer in its most common form. And there's some numbers here, but it's a terrible disease. At that time, 1984, the only drug that was approved is this one, BCNU. And he and I talked about this idea that maybe we could give a new kind of paradigm, which I'll call local chemotherapy. What if you allowed the neurosurgeon to operate on the patient, take as much of the tumor out as possible, but then before closing up the patient, line the cavity with little wafers that would deliver the drug BCNU. Now, normally BCNU has got a 12-minute lifetime, but if you put it in this little polymer wafer, it's protected. And also, you get delivery locally, right, to the brain, not all over the body where it usually could cause harm. So at any rate, this was our idea. But when you get an idea, you have to raise money. So we wrote grants, usually to the NIH, National Institutes of Health. And we did terrible. I thought I saved a lot of these. So in 1981, when we first wrote it, the reviewer said, well, the materials can't be synthesized. But I had some great students, and we synthesized it. Sent it back. The reviewer said, well, okay, that's good, but it's going to react with whatever drug you put in because of the bonds we used. So we sent it back again, and we showed that it didn't happen, and we sent it back again, and the reviewer said, well, but the materials will break in the body. They're what we call low molecular weight. They're fragile. But I had another couple postdocs, and they actually figured out ways to make these really strong, so there was no breakage. Sent it back again, and the reviewer said, well, nobody, takes new materials into patients much, or even animals, it's certainly going to be toxic. But we showed that, again, some of my students showed that it wasn't the least bit toxic in animals. Then we sent it back again, and the reviewer said it wouldn't diffuse far enough to kill the tumor. But again, one of our students showed that it did diffuse far enough. Anyhow, this kept going on until 1996 when the FDA approved it. It was the first time, actually, they approved a new treatment for brain cancer in 20 years. First time they ever approved this idea of local chemotherapy. All those students that I mentioned end up getting fantastic jobs, CEOs of multi-billion-dollar companies, department heads, deans. I think every one of them has been elected to one of the U.S. National Academies. The reviewers, they didn't do so well. I do want to show you what this looks like, but if anybody's squeamish and doesn't like the sight of blood, don't look. But here is what happens. There are little wafers going into the brain. Usually you put 6 or so in and close the brain. It's very hard for me sometimes to get good advice when I give a talk. But about 20 years ago, I was giving a talk at MIT on this, and my wife, Laura, came. And I asked her at the end, I said, well, what did you think? So she said, well, Bob, the talk was okay. That, by the way, is incredibly high praise. But she said, you had those 2 bloody slides on for 10 minutes going over every detail of the operation. She said, I don't know if you were looking, but all those chemical engineers were turning green. So ever after that, I did what I did today. I gave a warning and showed them very quickly. But, about 2 years after that, I got asked to give this dinner speech, actually at the Boston Aquarium, but it's to a group of neurosurgeons. And I told them the same thing. I gave them the same warning. At the end of the dinner, quite a number of neurosurgeons came up to me and they said, those 2 bloody slides you showed? I said, yes. They said, you could have left those on as long as you wanted. They said, those were fine. But those chemical formulas, they said, to leave those on for a neurosurgeon, they said it was just awful. So at any rate, if you look at the clinical data, actually this one came from Sweden and Finland. There's no cure, but it did allow you to prolong life, and you see a much greater percentage survival. This was from a 1997 paper, but even later, when you look at over 60 clinical trials, you see statistically significant survival. And this has now been approved in over 30 countries and used for the last 28 years. But also what it did is it really would introduce this idea of local chemotherapy. And that's been used, including our patents on that as well, to create, like for heart disease. One of the most common treatments people may know for heart disease today is what's called a stent, a drug. But what happens is, here's a stent. It's like a Chinese finger puzzle. But what happens is when you put these in to open the vessel up, about 50% of the time you get a problem called restenosis, which is the smooth muscle cells proliferate. And so what you want to do is have an anti-proliferative agent, again, an anti-cancer agent. In this case, people have used Taxol, but if you give Taxol systemically, the amount you need would kill somebody. So what they do is they take an ultra-thin, almost nano coating around the stent, put the Taxol in, and it locally delivers it. It's actually 1/1000th the total dose, but locally it's very high. And these are used by millions of people every year. So that was one example that I wanted to go over. Let me go over a second, which is the idea of gene medicine, some of which you've heard about today. And if you and I think Professor Sang, our last speaker, my colleague, I think made it very clear that delivery is a big issue. And here are different quotes from people, including my colleague Phil Sharp, who won a Nobel Prize for his great work in physiology and medicine. But the concern is solving the delivery problem. And one of the things, there's actually, going back to that Nature paper that we wrote in 1976, it shows that you can deliver, even though people told us we couldn't, molecules the size of RNA or even DNA. But even when you make these particles really small, sometimes there's other issues that come up. One is that they can aggregate and stick together because they're so tiny. Another is that macrophages can eat them. So one of the things that we did with Vladimir Torchlin and others is come up with coatings of polyethylene glycol. And if you do that, we could make, by adapting our manufacturing procedures, tiny particles. And just to prove that, in a paper we wrote in Science 30 years ago, you can show that by atomic force microscopy, by quasi-elastic light scattering. But what's really important, to back up what I was saying, is that if you put an orange dye in them, and then you look at what happens in vitro even after an hour, if you go to the upper left and you look at the thing without PEG, look at all those orange dots. Whereas the three different designs we did here, on the other than that, there's either no orange dots or very, very few. So that actually would help solve that problem. But there are other problems that would also occur. One of the things, particularly if you want to encapsulate a nucleic acid, you want to actually encapsulate it with something that has an opposite charge so that it will sort of be small and condensed. But when you do that, and in this case you want to encapsulate with something cationic, that can cause toxicity problems and what's called complement activation. So one of the ways that Francis Arnold sent me a student, Dan Pack, a number of years ago, and he, along with Dave Putnam, came up with the idea of attaching something to a molecule. And people had done some of these things before, but never for a purpose like this, that would make it neutral at pH 7, for body pH, but when it into the cell, in particular into the endosome, because of its pK, it would become charged. And that meant because it was neutral at body pH, it didn't cause a toxicity. And when it was charged, it enabled you, as Fang was talking about in the last lecture, to break out of the endosome. So, and then there's been great work also done at Vancouver on this issue as well. But basically, this is just the idea of the lipid nanoparticle that we're talking about. And so you have cholesterol, the PEG lipid, which I mentioned, the structural lipid, but then this ionizable cationic lipid. And those, the Pack paper and actually the Vancouver paper are listed on the bottom. And so I thought I would just show you a little bit more on this. You could even put targeting molecules on it to target it possibly to specific cells in the body. And since I realized I didn't explain this that well, I was very fortunate a number of years ago that Nova, the TV show in America that has some things on PBS, they were making a TV show called Making Things Smaller. And they came to MIT and they filmed some of the stuff we did in the lab. But then they made a video, and I liked the video, so I'm going to show you it.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
So I should tell you that the clinicians I work with tell me it doesn't really blow the cell up quite like that. But you get the idea. And one of the exciting things about this is now maybe you could deliver siRNA with it and or mRNA. But again, this so I thought I'd tell you a story. So if you, and this is a story of messenger RNA. If you go back to 1990, there's a paper in Science by Wolf and others showing that you could actually give messenger RNA to an animal and get expression in the animal and get proteins in the body. But you also got some immune effects. And that was an unsolved problem until 2005 when Caitlin Carrico and Drew Weissman at Penn worked out ways to modify it, for which they received the Nobel Prize in Medicine and Physiology last year. They also wrote a patent, and they tried to start a company, but they couldn't get anybody to fund them, as good as that work was. About 5 years later, Derek Rossi, a scientist at Harvard, was using these kinds of approaches to change a regular cell into an iPS cell, which is a stem-like cell. But rather than use viral vectors, he used messenger RNA. And he also wanted to start a company, and he went to really good places in the Boston area like Third Rock, and they all told him again, this can't work, it's never, especially delivery and other things. So Tim Springer told him he should come see me, and I thought when we talked that we could hopefully solve this delivery problem and maybe even get funding. And I thought that because, we had spent by this time 36 years working on this. Again, it's academic work, but I thought there was enough things that we'd shown that people said you couldn't do, that maybe with a company we could do it. So with Derek and Ken Chen, who later would go to the Karolinska, and Nubar Fayan, who worked at Flagship Ventures, we started Moderna. And the idea is that, the idea is that, and this is just a central dogma, but as you heard eloquently this morning from Greg Winter, that proteins have been fantastic drugs, but they're not always so easy to make. And in particular, let's take a vaccine. If you took flu vaccine, you might want to grow up eggs, and you might want to start over a year in advance or more so that you could make enough vaccine. So that means, let's just take flu, you're going to guess. Are you going after H1N1, H1N2, et cetera? And if you do that, you might guess wrong. And flu vaccines, maybe 40 or 50% effective because of that. But the beauty of messenger RNA, if you used it, and let's say this would really work, you put it in a nanoparticle, inject it into the body. This is all theoretical at the time, that basically the body would be the manufacturing plant, and it would do exactly what you did outside the body in the other case. So that was really our idea. And also, we thought that you could maybe treat other diseases that you might not have been able to treat before, like to make membrane-bound proteins or intracellular proteins. So we started the company, and and it got widely criticized by the press, by different journals and stock analysts. But at any rate, by 2019, and a number of our students worked there, it's right across the street from my lab, but by 2019, we had a number of things in clinical trials, early stage, and then COVID hit. And I wanted to show you what the Moderna scientists did. So on January 11th, 2020, the Chinese scientists published this genetic sequence of what's called the spike protein. And it took less than 2 days. In fact, it took about 10 minutes to design the vaccine. But they kept repeating it to make sure it was right. Then put it in nanoparticles, ship it to our clinical partner, the NIH, for testing. And the first patients were treated in March. And they did phase 1 trial, phase 2 trials, ultimately tested over 30,000 patients. And of course, I think people know that it's been approved all over the world. Just to give you some data on this, on efficacy. Here you compare vaccine, unvaccinated, vaccinated with no booster, vaccinated with a booster. if you look at effectiveness at the Mayo Clinic, there are really only 2 vaccines in the U.S. that really were ended up being used. The Moderna one, which was 94%, and the Pfizer-BioNTech one, which is 91%. Both of those, and these are the only 2 messenger RNA vaccine ones. A little bit more, this is a more recent study just from last year in JAMA, over 6 million patients, showing that it's the most effective mRNA vaccine for older adults like me, 76 or so. I'll give you some more. One of the things I think that always comes up, and especially in today's world, is it safe? And I think it's really important to, and I'm sure any clinician will tell you, nothing is perfectly safe. But if you talk to different health agencies, they might say something like that vaccines are the most effective public health tool ever, ever developed or ever conceived. So I think you always have to look at what I'll call the risk-reward ratio. One of the reasons this would get approved is that a lot of people were going to die and did die. So you have a choice maybe between death or debilitation or taking the vaccine. Now, a good question I think to ask is how would the mRNA vaccine compare safety-wise to a well-established vaccine like like a flu vaccine? And that's been studied as well. I'll just pick one example, which shows here that if you look at safety profiles, you get a lower risk of serious adverse events if you use mRNA vaccines compared to flu vaccines. And I thought I would just give you some more numbers. This is over just an 18-month period in the US, but there were 49 failed vaccines and 2 approved, 209 failed treatments and 1 approved, 153 failed antivirals and 1 approved. And just to go a little further, if you look at this again over that 18-month period, there are foundations that have estimated that it prevented 3 million deaths, prevented over 18 million hospitalizations, prevented almost 120 million COVID infections, and saved the U.S. $1.5 trillion. And there are other things, of course. This is just one well-known example that people know. What you may not know is that it could also be used for many other things. And I was just going to give you one other example, which is cancer. And here is the what's called the individualized neoantigen therapy. It's also been called personalized cancer vaccines. And what was done is to take patients with advanced melanoma, and now what they're doing is selecting up to 34 mutations known as neoepitopes in a patient's cancer cell. So it's going to be a very personal vaccine. You incorporate the genetic code of those neoepitopes into an mRNA vaccine. It's exactly the way you just saw. Rather than going after the spike protein, you're doing the 34 neoepitopes. So mRNA and nanoparticles. And again, you heard from Greg this morning, Dr. Winter, that Keytruda is the #1 best-selling drug right now in the world. And what was done, and it works very well. That's the reason why they use it. So in this trial, half of the patients got Keytruda and half get Keytruda plus personalized cancer vaccine or this individualized therapy. And it's what's called a double-blind study. That's the way all these are done. So they don't know what's going to work until you break the code. But at the end of 2 years, the risk of recurrence or death was reduced by 44%. At 3 years, 49%. So this is what's called the phase 2 trial. Now it's in a phase 3 trial. So I thought I'd just end with a couple of future points. This is where we are now. But, about 12 years ago, I had a visitor who was a specialist in the internet and computer things. And he and his wife had this foundation, and they were interested in trying to do things for the developing world. I'll just show you a picture in my office. That's me on the left and the computer person on the right, which is Bill Gates. And one of the things that he was interested in was people in the developing world don't come back for a second injection most of the time. And so could we ever come up with a way to solve that problem and maybe come up with something that looks a pattern like this? So you get a pulse at time 0, but also maybe at 1 month, 2 months, and so forth. And the idea that we had is that basically you could put the vaccine or whatever, make that the red dot, and cover it with a material. And that material could be a polymer. In fact, we used a FDA-approved polymer, lactic glycolic acid, which breaks down into water and CO2. And the idea, think of these as little eggshells. What if we could make them break at specified times? And so we actually did that. Basically, here's the model of polylactic acid. Again, if the blue part would be a simulated vaccine. But here, which I was very excited about, we had published some of this in Science, is that you can get one injection, this is in an animal, and 11 separate pulses. And we're actually going even further. We're collaborating with one of my other students, Mark Prausnitz, who's at Georgia Tech, and he is an expert on what I'll call microneedle patches. And so what you can do, if you look at here, you see a SEM of these patches, and basically they don't go in very deep in the skin, so it doesn't really hurt much, but they go in deep enough so that you could deliver these tiny little particles. So hopefully someday we'll be able to take those patches that you see over on your far left and put them on patients and hopefully ship them all over the world. We've also made 3D printers that can make these pretty well. So I thought I'd end on a totally different materials note, and that is, could you even use materials to help me make someday new tissues or organs? And here we're motivated by little babies like this who are dying of liver failure. That's Jay Vacanti is one of my collaborators, and he treated that little boy. And the only way a little boy like that would live is somebody else has to die, and then he'd do a transplant. And so he asked me one day, could we ever come up with a way to create new tissues and organs from scratch? And I'm going to vastly oversimplify this, but it's actually a lot's been done over the last 40 years. And the idea is that maybe you could take any cell type, including stem cells. If you inject them at random, not much happens. But cells can be smart, and if you put them on the right scaffold under the right, what I'll call reactor conditions, they can actually grow on these materials and theoretically make any tissue or organ. This is from a paper in Science that's now actually been cited over 9,000 times. And from a material standpoint, we would design, again, degradable materials that you could put these on the scaffold. Again, if it was, you might need to immunosuppress the patient, but you do that anyhow if you're doing liver transplants. But it may depend on the tissue you're doing. We also synthesize brand new polymers because some cells will not adhere very well to polylactic glycolic acid. So the one on the bottom, which we published in JACS, you can take just substitute lysine, and then through that NH2 group, you could do certain reactions where you can link specific amino acid sequences. So let me just show you two quick examples of what you can do and what was done. So let's say somebody's burned. So this is real life example. Oh, I'm sorry, this is just a scanning electron micrograph of the scaffold. So now the little boy who's burned. And this is, again, a true story. So he's burned, and what you can actually do is take neonatal skin fibroblasts, put them on this scaffold, put them on the child at the time of the injury. Let's come back 3 weeks later, and now 6 months later, and he's pretty much healed. And not only could you do that, but you could actually someday, I believe, make organs or tissue on a chip, which someday might reduce animal or human testing. And to that end, here's work that Gornata Vunyak and Milika Ritzik, who were in my lab, did with a heart, and actually even had a company to do this. So you can take heart muscle cells, and you get this characteristic structure. But if you, and I'm going to show you a video, if you put 6 million cells on one of these scaffolds, let's just look at it. And we're not shaking this or anything. You can just see it beating like a real heart. I hope people can see that. And you can even take an EKG. And in fact, you can make many of these. And I just thought on the final slide, this is what we and our students are doing and trying to make almost every tissue. And I won't go through all of them, but You see Julie Andrews up on the top. We're actually been working with her voice surgeon, Steve Zeitels, to make new vocal cords. On blood vessels, actually, that's been really exciting. Laura Nicholson, who was one of my fellows, she was a full professor at Yale, left to start a company called Humacyte, and they're already treating soldiers in the Ukraine who are dying of making her vessels, and it's at the FDA, and many, many others. So I want to thank you for listening to my struggles, but also my dreams. And I hope that what I've tried to go over is how chemistry and engineering, can at least maybe take us a step forward in relieving suffering and prolonging life. Thank you so much.